Hi, this is Alex from devstories.eu. AlphaFold, Protein Structure Prediction, Episode 3 of 21. The most powerful feature of an AI model is not always its output, but its ability to tell you exactly when it is guessing. If you look at a predicted protein model and assume every loop and domain is perfectly placed in reality, your downstream experiments will likely fail. To actually use these models safely, you need to understand evaluating predictions, pLDDT and PAE. AlphaFold outputs two distinct metrics to quantify its uncertainty. The first is pLDDT, which stands for Predicted Local Distance Difference Test. This evaluates local confidence. For every single amino acid residue in the protein, AlphaFold assigns a score between 0 and 100. This score tells you how confident the network is in the local backbone structure. When a residue scores above 90, you are looking at very high confidence. At this level, even the side chain orientations are usually reliable. A score between 70 and 90 still represents a good, trustworthy backbone prediction. As the score drops below 70, confidence gets shaky. Here is the key insight. When you see a pLDDT score below 50, your first instinct might be that the model failed to find the right fold. That is usually incorrect. A very low pLDDT score often indicates an intrinsically disordered region. The protein physically lacks a fixed structure in isolation. It might be a flexible linker or a tail that only folds when it binds to another molecule. The AI is not failing, it is accurately predicting that this piece of the protein is naturally floppy. While pLDDT is excellent for local folds, it has a major blind spot. It evaluates regions in isolation. Consider a protein with two completely separate structural domains connected by a long string of amino acids. You run the prediction, check the pLDDT, and see that both domains score above 90. Their internal structures are solid. However, pLDDT cannot tell you if those two domains are correctly positioned relative to each other in three-dimensional space. To solve this, AlphaFold provides a second metric called Predicted Aligned Error, or PAE. This metric evaluates the relative position of domains. PAE measures the expected distance error between any two specific residues in the protein. The logic asks a simple question. If we align the prediction to the true structure perfectly on residue X, how many angstroms off will residue Y be? This gives you a two-dimensional grid of pairwise errors. Going back to the two-domain protein scenario, if you check the PAE between residues inside the first domain, the error will be very low. The same applies inside the second domain. But if you check the PAE between a residue in domain 1 and a residue in domain 2, the error might be very high. A high PAE between domains means their relative orientation is completely uncertain. The model knows exactly what the two separate shapes look like, but it has no idea what angle they sit at relative to one another. You must evaluate both metrics together to understand the full picture. You use pLDDT to trust the local folding and to identify regions of natural disorder. You rely on PAE to verify if the overall global arrangement of those distinct folded parts is actually fixed or just a spatial guess by the algorithm. Never treat a predicted structure as a single rigid truth. Treat it as a map of probabilities where every domain and linker carries its own proof of reliability. Thanks for spending a few minutes with me. Until next time, take it easy.